Hey everybody, and welcome back to this episode of Tales of the Resistance. I'm Mara Zultz. I am a staff member with the I Am Responsible team. We talk all about antimicrobial resistance, what it is, where you can find it, how it impacts our lives, and what we can do about it. And in this current small series of podcasts, we are interviewing a set of environmental science researchers at several different universities across the country who are all working together on a single project, which they call COMRADES, or Connecting Antimicrobial Resistance, Agricultural Decisions, and Environmental Systems. The overall goals of this project are looking at antimicrobial resistance, many different types of measures of antimicrobial resistance in surface water streams, in agricultural settings, and then try to use different modeling practices and risk assessment practices to understand how the presence of antimicrobial resistance in those far out rural areas has an impact on human health. So in this particular episode of the series, we're going to be talking with our risk assessors. So we're going to go and we're going to take it a step further from the sort of chemical components and microbial components that we've been exploring and predicting their movement in environmental settings to try and understand the connection to human health. And that's where this risk assessment part comes in. So I'm going to go ahead and transition us over there to our previously recorded interview with Dr. Bing Wang and her graduate student Jaber who's a PhD student at the University of Nebraska-Lincoln. Thanks for joining us for a little while here and giving us some of your time. But we always like to start everybody to kind of give us a sense of your background, how you got into the field, why you chose AMR, kind of how you got here. What's your brief history of AMR research?
Okay, I'm Jaber. I'm a PhD candidate in the field of food science and technology at UNL. I started my PhD back in 2022 with Dr. Wang. Before that, I did my bachelor's in microbiology and also my master's in medical microbiology back in Iran. Well, during my master, I became more exposed to AMR while I was working on the project that were mainly focused on detecting pathogens, resistant pathogens from hospital-acquired pneumonia patients. And that time I realized how big this problem is and how patients die so fast within just a couple of days, and physicians cannot do anything about that when they get to the point that they get resistant infection. So I was really interested in working in this field, and then I became more because, as you know, antibiotic resistance is not just a healthcare problem. It's something interconnected between environment, agriculture, and even industry, everything. So joining Dr. Wang's lab gave me this opportunity to work on this project that is called COMRADES, and it's basically help us to work with other scientists in different fields, environmental scientists, veterinary, food safety. Dr. Wang is from the veterinarian part, but we all working together to find a solution for this problem. And so that was very, I think, good switch for me.
Yeah. So my training is mainly focused on veterinary medicine, as Jaber mentioned. So as a veterinarian, it's a very common thing for us to think about how to use antibiotics in a prudent way and how to avoid antimicrobial resistance. And when it comes to the UNL, and my job is mainly about research, teaching, and extension around risk assessment, which is to use some data analysis techniques to support the public health decision making. And one of the topics that we have applied as a major one in our group is about antimicrobial resistance. And it's mainly about the impact on the human infections. That's a major component, but also it can touch on the environmental health and at the same time thinking about the protection of animal health at the same time.
Have you ever had an experience with AMR outside of the academic side, like in your personal, have you known someone? Had you ever heard about AMR before you got involved with it in academia?
I don't exactly remember, but I know that during COVID era, I remember like maybe someone from our far relatives died because of the resistant infection. I mean, you get the COVID, of course you have all the thing, but usually patients who went to hospital and hospitalized, sometimes they develop the resistant infection after being hospitalized, and then unfortunately they don't die because of the COVID, because of the resistant infections.
Well, and I think I picked up one word from your first discussion about your rationale for this, and that you talked about connections. How the thing that's going on in the environment is impacting this human health, and how quickly that those changes can happen. And I think one of the things that has been coming up in our other interviews with folks is you guys have been kind of discussed in all of those as like, they're the connections people. They're the people who are going to put this all together and like give us some meaning, about what it all means. So I guess we've had a lot of anticipation to get to talk to you guys about what are the connections being made in this sort of environmental to human health pathway. But probably before we get to like what those connections are, we probably need to hear about like kind of what the process is, a little bit about what your guys are doing that can actually make those connections possible.
In this project, I think what we can do is really follow the steps of a risk assessment. A very important step in risk assessment is called exposure assessment. We're trying to make the linkage from the source of antimicrobial resistance, either bacteria or genes, all the way to the exposure of human. So like in this project, we proposed 3 different possible pathways that the people can expose to the antimicrobial resistant bacteria and the genes that originated from the agriculture activities. So they can release from, for example, the runoff of livestock place into the water body. So that's the starting point. But how people have the contact with those bacteria and the genes, that could be first through, if the people use the river water as a entertainment activity location, so swimming, accidental swallowing some river waters, that could be one way. And another surface water could be the input for the drinking water treatment plant. So out of that, the drinking water could be another way that people can expose it to if still some bacteria and the genes maintain in the drinking water even after the treatment. So the third one is through the irrigation water. So the water can be used to irrigate the fresh produce, especially those with very minimum process, that there's no killing step, there's no further step to remove the bacteria or genes. Then consume those fresh produce. It could be another way that people can make the exposure to those resistant bacteria and the genes. So that's our work. So we make the connection from the discharge site all the way to the human exposure and predict the impact of a human risk. So I think that's how our colleagues mentioned about that to combine everything together, because our collaborators, they do have the samples collected from the field and from the discharge site, and maybe along the river, they also have different points of data collection. And our job is to make the chain even further down to the human exposure. Yeah.
There are so many different routes and pathway that human can be exposed to, like Dr. Wang also mentioned those. But what we discussed here more was focused on water. But for sure, there are different other pathways that human can be exposed to. Water has the capacity that you can have these antibiotics that are in this water environment can put a pressure on selection of antibiotic-resistant bacteria, and it also can transfer genes between each other. And this way, they actually, even non-resistant bacteria can become resistant, and therefore this can pose a threat.
As Jaber mentioned, here in this project, we mainly focus on the water as a carrier of those resistant bacteria and the genes. So take one of the pathway that I mentioned as example through the irrigation water. So the water from the surface water, if they used as the irrigation water, so the water depends on the irrigation approach. If there's a sprayed overhead, then that can be attached down to the surface of the fresh produce, such as lettuce, right? But, during the growth period, and for the bacteria and the genes, it won't stay over there all the time on the surface. It may decay. So under different temperature, over different time, some decay rate may apply. And also bacteria may grow. So if there's a resistant bacteria under suitable temperatures, and the population of the resistant bacteria may grow bigger. So considering in all the way to along the road, so after the harvest, during the transportation, and again, decay and growth may occur for bacteria. And also, if there is a washing step and the physical removal may be considered. So all of those details along the supply chain are the factors that we need to consider about the change in the population of the bacteria and also the genes. So that's through the mathematical model, and we collect the information from our collaborators and also using tools like a systematic review and meta-analysis to collect the information from existing publications to really provide the value for the variables that are needed in the model. So that's how we build a chain model. By doing this, if you change some parameters at the upper string, then you can predict what's going to happen to the human infection or health risks. So that's the reason that why we do the risk assessment chain model approach, because it will help you to predict if you do some interventions. For example, if you use a different landscaping approach or land use for the agriculture site, maybe it will help to hold and reduce the release of antimicrobial resistant bacteria and the genes into the water, and all the way reduce the human exposure through the irrigation water route.
How, this is really interesting. How accurate would you say the chain model is in predicting, let's say you add prairie strips, and you want to see how adding a prairie strip will affect the risk to the human health. What kind of accuracy level are you looking at?
That's actually a very professional question.
Yes.
So for any model, uncertainty will be involved. So for example, the uncertainty about the model structure is one of the components. By saying that, what I mean is your model really representative of what is happening in the reality? So it's for us as a risk assessor, we are not only sitting in front of the computer and just imagine what is happening there. We actually work with the experts to really know about those details along the supply chain. And communicate the way that we built the model with them and listen to their feedback to see, maybe they say, no, here you need to also think about some other factors. Then we modify our model that way. So that's the first component about the uncertainty that might be involved. And the second is the data inputs. As I mentioned, so the model is actually heavily rely on the data. A lot of time in real world situation, variance is a big thing. So some people, they may only rely on one single study to come up the values for a parameter. But why is that paper, not the other paper, right? So we do the comprehensive systematic review approach to capture the relevant data as much as possible. At the same time, we also use risk of bias tools to evaluate the quality of the data that we use to put into the model. So that's the way that we can control the uncertainties that we may introduce into the model development to make sure that the accuracy is as high as possible. At the same time, we do have a way to do the validation of the model results. So, for example, the model can predict what would be the concentration of a resistant bacteria or genes on the surface of a lettuce at the time of harvest or at the time of retail. If we also have empirical data collected, like in the United States, then we can compare our predicted value with the observed value to see if our prediction match with what is happening over there based on what is the system that we're modeling. So, Bethany, answering your question first, we build the model and trying to reduce the uncertainty as much as possible. And also we have a way to know that if our model is validated.
Mm-hmm. So like when you have different pathways, so somebody is swimming in the water, that's one pathway to the possibility of exposure to and risk. And then maybe another one is putting the irrigation water on lettuce. And then another pathway you mentioned was wastewater treatment facilities not able to remove it. So these different pathways, what's the severity of risk like between them? Is one a lot higher risk and one a lot lower?
That's a very technical question. So for sure, what you're doing is to answer these questions. And let me just go back a little bit and explain about risk assessment. For the risk assessment, we have hazard characterization and also exposure assessment. So in the hazard characterization, of course, our hazards here in our study, we're talking about the genes, and we know that there are thousands of resistant genes. It is in a conventional risk assessment method, usually we have the dose response model, meaning that how much of a hazard or a resistant gene your body needs to take to have a harmful impact. But unfortunately, there is not something like a dose response model for resistant genes. What we did here, we were actually defined some criteria, and then based on these criteria, we extracted all the data that were available and asking experts to weight those criteria, and finally we came up with a score for each specific gene. These scores kind of representing how dangerous or risky a gene can be for human. And then later we have the exposure assessment. We want to know how much human can be exposed to these genes through different routes. Or like you said, we have swimming, we have maybe drinking. That's actually the part that we are now focusing on. We want to know, for example, if a sample of water that we have that contains this quantity of the gene, this concentration, and contain these specific type of genes that we have the score for, and this water is going through the treatment plant, and we have different stages of treatment, then what is the concentration on the tap water, right? That's the whole model. That's the whole change. Mm-hmm. You have different stage if you want to see that okay, this is the log reduction for each of the stages of treatment plan. And then at the end, when you're drinking a cup of water, you're gonna evaluate based on the quantity of the gene that you have and also the risk that we actually defined in the previous step. So that's the question that we are now working on, and hopefully we are gonna be able to answer this question at the end of when this study or this project is basically done and get to that point.
And I'm sure that like the reason that it's in progress, you don't know it yet, is the complexity of the pathway. Like there's, like you've mentioned before, things that you didn't know, and you're talking to experts about what components to be added to the pathway model, the chain model. I guess I'm a little curious about if there's anything that has popped up either in this study or Dr. Wang can speak to previous studies that was like you were working on a model like this and you kind of got thrown for a loop, something you really did not expect to need to include in one of these to understand how the pathway actually functions.
Yeah, I would say one of the factors that make this project is a little bit more challenging compared to the other risk assessment project that we have been doing in our group is because here we focus on the genes. So Jaber explained that a little bit, but I want to go further a little bit. So one of the unique or innovation that's involved in this project is about the risk scores we developed for different resistant genes. So traditionally, as Jaber mentioned, there is different steps of risk assessment. The hazard characterization is the step to link between the dose and the probability of some adverse health effect going to happen. So for example, if you take salmonella from the consumption of a poultry product, it depends on how many numbers of salmonella cells you intake. The probability of having salmonella infection is different, right? So from the exposure assessment, that prediction can tell you how many copies of genes or how many resistant bacteria that the people can be exposed to through different routes. So the hazard characterization part is to, okay, if you give me the dose, then I can predict the probability of your risk. So that's what is very important about the hazard characterization. But for the resistant genes, there's no existing hazard characterization in this field for risk assessment for antimicrobial resistance at all. The reason is if you only think about the gene itself, if it's not embedded into a pathogen, it doesn't really show the infection. So for example, if this gene is carried by a commensal bacteria, which is not pathogenic, if not assuming that the resistant genes can be horizontally transferred to the pathogens within the gut of the hosts, then probably there's no infection at all, right? So the gene itself, it doesn't really necessary to pose a risk assessment. But here, Jaber's work is we can to imply the potential human health risk based on the diversity of different genes that the people exposed and the quantity of the gene people exposed. So I think that's the biggest innovation of this project. So that's also pose a lot of challenges for us to do because there's a very limited prior experience that we can learn from. This is really another advancement of the risk assessment methodology. So back to your question, Mara, is not a surprise. And when we proposed this study, we already know this is a big challenge. But I think it is very important. A lot of time, so first is a lot of time when we work with environmental scientists, they measure resistance in a different matrix as the food scientists. Food scientists may measure resistant bacteria, but environmental scientists more likely to measure resistome, which means the composition of different genes in the environmental samples without figuring out what type of bacteria this gene is actually embedded in, right? So this is because of the challenge in the data type, the type of data that we get. So we have to make an advancement of the risk assessment methodology part to meet the needs for risk assessment for antimicrobial resistance if this discharge or originated from environment. And also, I would say a lot of time, once we make this updated, we call it the next generation risk assessment, the application is actually more, it will be broader. So, for example, people can use more advanced metagenomics approach, just looking at the composition of the genes and maybe the high throughput qPCR to know the composition of the genes and at the same time to quantify those genes with that information, and we will be able to give you a prediction about the risk scores. So the thing is, risk score maybe doesn't mean too much, but if you have the comparison, like if with and without the intervention, or before and after intervention, then I can tell you, okay, if this intervention really is working or not. So in terms of the massive surveillance program and to use the surveillance data to evaluate the intervention efficacy, I think this advanced approach will be very helpful.
Yeah, I noticed that in your conversation that we were talking about genes, and last time we talked about this several years ago, that was not on the table because it wasn't possible. So big step forward for the risk assessment approach, and yay for Jaber's work.
I think I never talked to people about AMR, but I would love to. That's not my...
Well, you are right now. You're doing it.
But I think one thing that I always say that, so it's the AMR, like at the beginning I said that it's something that is interconnected between everything, like environment, human, animal. And so everyone is responsible. If you're a farmer or if you're a physician prescribing antibodies, or if you're a veterinarian, or if you're, I don't know, if you're even a scientist, everyone is responsible for that. And this is a very big challenge that nowadays we need to find a solution. And WHO announced that every scientist in different field should take an action toward this problem. So I would say that do as much as you can do to just maybe using less antibiotics and more using, I mean, less antibiotics to just avoid resistance, because it's very hard if you get to point that even sometimes you have the antibiotics residues inside the environment, and that can also pose pressure. But even these resistant genes, they can last longer even after you remove the impact of the presence of antibiotics. So even downstream water or environment can be impacted by what we are doing now and even later.
Yeah, I would say because we do the chain model, right? So we can connect everything. So we do have the opportunity to see along the way and what can be done. If that can be done, then what would be the efficacy of reducing human risk? And we do observe that everybody along the line, if you do something that help to reduce the risk, that's from the prediction. So scientifically, that's supported. So yeah, back to Jaber. I just want to support that Jaber's mission that's everybody, don't pointing fingers to the others. All the sectors needs to work together to combat antimicrobial resistance.
Yeah, for sure. I was going to ask, what's been your favorite, most enjoyable part of doing this research? What have you liked the most during this project?
I can say that so there's so many things that I like about this project, but first is the ultimate goal of this project, which is helping public health, and it helps that at the end, if even we can make a really small change in fixing this problem, then I think I would be so happy. So not only public health, but even animals can get sick due to the resistant pathogens, or so all together improving the environment, our environment, also animals and public health. This is a very I'm so excited about this project, and I am a part of this project, so it's very fascinating. But also working along with people from other universities and collaborators from different fields, and learning how they look at this at the AMR from their aspects, from their views. It's very interesting because when you talk with, I come from a kind of a clinical background, and then you see that people like working environmental scientists or people from the point of water safety, all from different fields, and then you sit on the same table and you talk. That's very interesting, that how people see AMR from their point of views, and then we can find a way to discuss and find a solution for that. So that's very interesting for me.
Yeah.
I like it.
Okay, inspirational.
Yeah, the part I like the most actually just came out to my mind is I love to work with my lovely, lovely, lovely students and to watch them to grow. And to look at Jaber's responses and into this discussion today, it's not only academic growth, right? During the past few years, they grow in vision, in mission, in responsibility. I think that's the part that, as an advisor, I enjoy the most.
Yeah. Awesome.
Thank you. Yeah, I enjoyed working. I also enjoy working with my advisor, Dr. Wang. It's very
Yeah, better include that. We'll make sure we put it in. We'll put it in the podcast for sure. Well, we're wrapping up time, so I want to make sure I leave a little bit of space just in case we skipped over something you want to be sure we include about your process, your learning.
I can say I am so excited about the future of what you are working on, and I think it's gonna be promising, especially with the advantage of rapid detection methods that currently we are using, our collaborators are using to detect AMRs and genes and also bacteria in the field, and also this kind of collaborations that we are working on, hopefully. And I'm 100% sure that we can make changes. But yeah, I'm so excited about the future, and I hope that we can do something all together to just a little bit mitigate the problem of AMR.
I'm excited to be able to share your guys' results and see what they are for myself, too.
Yeah, this is always the highlight for me, is the risk assessment discussion, because it does bring everything together. And so I've always gotten really excited every time Dr. Wang is sharing her results. Oh, I see all the stuff that I did. Now I see the meaning behind it in big picture stuff. So, well, it was really great to take this little time with you and get to chat, and we'll probably chat some more in the future too, but to take this time and get to hear what you've been working on, at least at this sort of intro level, and getting to meet you and everything. So thank you so much.
Yes.
Yeah, thank you,
so
Mara.
much.
Thank you, Beth.
It's a pleasure
Thank
meeting
you.
you.